So this is an overview of the regulation of translation using RNA interference, or RNAi. So ultimately what the aim of this is a piece of mRNA has been produced, but let's say the cell doesn't want that piece of mRNA to be translated into a protein. So it's already been transcribed, but it doesn't want to produce a protein, doesn't want to reach a ribosome. So one way that a cell can regulate this and prevent it from happening is using RNA interference. The process starts off with some double-stranded RNA. An enzyme called dicer splits that double-stranded RNA into into smaller fragments. So these are small interfering RNA fragments, and they're double-stranded at this point. Small interfering RNA becomes single-stranded, and it associates with enzymes to form RISC, which is RNA-induced silencing complex. So it's a mixture of enzymes and single-stranded siRNA. And what's important here is that these bases will be complementary to the mRNA molecule, bases on the mRNA molecule that you don't want to be translated, that you don't want to reach the ribosome. So what happens then is the siRNA, the small interfering RNA, inside the RISC complex, complementary base pairs with the piece of mRNA, and then what happens is the mRNA is cleaved, it's hydrolyzed, it's broken within the RISC complex. That mRNA is no longer functional, and it cannot be translated into a protein. So this whole process is called RNA interference, or RNAi.